from San Jose, California, and brought to you by Think Tank Learning. Welcome to Meet the Experts. This is a forum in which we pick the brains of experts and professionals from various industries. We give students inspiration by showing them the academic journeys that they can imitate as they prepare to become experts themselves.
I'm your host, David Ichnuin, and today we're going to talk about drug design. Advances in molecular biology and computer science have allowed scientists to predict the structures of molecules that operate inside a cell. When these machines start to go wrong, the result is a disease or a disorder. Society is always seeking that magic pill that will cure ourselves and our loved ones. What if we could design a drug that works better for you than for anyone else? And what if that drug had no negative side effects? No nausea, no vomiting, no allergic reactions. Well, our special guest today is at the forefront of drug design. He is Russ Altman. Russ Altman is professor of bioengineering, genetics, and medicine at Stanford University. And by courtesy, he is also professor of computer science there. His research interests include methods of understanding drug action at the molecular, cellular, organism, and population levels. He studies how genetic variation affects the way that drugs work or don't work. He also studies how molecular machines operate and why they cause drugs to fail. Professor Altman, welcome to Meet the Experts.
Thanks very much. Thanks for having me, David.
Yeah, so to start, could you briefly describe the process of drug design?
Yes, it's a complicated, lengthy, and very expensive process. Many drugs cost $1 billion to create. So basically, there are discoveries early on that perhaps a molecule has some effect on the disease. Maybe they learn that the molecule is involved with the disease, and we realize that if we could only change how that molecule works, we might be able to the disease better. So that's the first idea. Then we do a lot of things to try to find small molecules. What I mean by small molecules is things that are roughly the size of a glucose molecule or an ATP molecule, or aspirin is a small molecule. And we try to find small molecules that can interact and reduce that protein. Usually it's a protein that is going to arrive. So then there's a process of optimizing it, testing it in animals, sure it helps them and doesn't hurt them. You start out with rats and mice, and then you might even move to rabbits and pigs. We don't usually do anything with monkeys because they're actually too close to humans. So then all the data looks good. You then introduce it into humans, healthy volunteers, just to make sure it's very safe and it doesn't do anything bad to them. Then you start exposing patients who have the disease just to find a dose, to see if it works. But what would be the dose that they would tolerate? What would be too much? What would be not enough? And then finally, and I'm compressing 7 or 8 years into 12 seconds here, finally, you do a trial of thousands of people with the disease against either a placebo, which is an inactive pill, or against the current best drug for treating that disease, and you do a head-to-head and you see how it performs. The FDA, the Food and Drug Administration, which is a U.S. agency, a government agency, they watch this process. If they find the data to be compelling, they allow you to market the drug for a specific disease based on all of this data that I just described.
Well, and so you said, so part of the process is an animal testing and experiments, of course, to protect humans. And so how important would you say is animal testing in drug design, right? There are people out there who are very against that, and
right,
it's a roadblock for them.
So it's a two-sided coin. It's critical because every now and then you have a molecule that looks like it's going to be great, and you give it to a mice or a rat or another animal, and there's a terrible response. Like the animal gets very sick, and basically it kills the drug. You say this is not going to go forward. Now, we feel bad for that mice or that rat. We really do. But think about if we had given it to a child or a man or a woman. So many of us consider it to be regrettable but important to do. On the other hand, that data is not perfect. And we can have examples of medications that look like they're going to be perfectly safe. They fly through all of the animal data, but when you give it to a human, it doesn't work as expected. So it's a two-edged coin. And as you know, I'm a computational person, and one of the things I'd like to do is to develop computational methods that, even if they don't totally replace the animal testing, will make the animal testing much more focused and at a smaller scale, because we have such good computational evidence that we can experiment on less animals.
Okay, so so how did people do drug design before computational techniques were involved?
Yeah, so, and I should say that the vast majority of drugs on the market today were not designed by computers, because the computers have been only really active in this for 10, 20 years. The way it usually goes is you have an experimental system that mimics the disease. So you either have that same disease in a mice or a rat, or even just in a cell. Like a cancer, you might have a cancer that, instead of being in the body, is just growing in a Petri dish. And then you do what we call a screen. You have a library of literally hundreds of thousands of molecules, and you pour a little bit of molecule on a whole bunch of Petri dishes, and you see if any of those molecules kill the cancer cell. The ones that kill the cancer cell, you then advance to animal testing. So it was very, what I would call, empirical. Try it out, see how it works. And therefore very expensive, because as you can imagine, setting up the robots, and in the old days they didn't even have robots, that would have, 100,000 Petri dishes, or at least little tiny test tubes, and then making a robot that's going to add different medications or potential medications to each of those test tubes, and then see if it works. That's extremely laborious. And so we're hoping that computational techniques will help that, but that's how it was done in the old days. You find a lead, and then you're off to the races. Then you send it to your chemist to figure out how they can synthesize it. Can they make it work a little better by adding some chemical groups or subtracting, just tweaking the molecule a little bit? And then you try to optimize for the effects you're seeking and optimize for the minimal side effects as possible.
Wow. Okay. And so if there's a new concept called personalized medicine, and if personalized medicine is defined as creating a drug that works just for you, right? Your genetic background, your biochemistry. What does drug design or computational drug design bring to this concept of personalized medicine?
So personalized medicine is very exciting. In some ways, the first version of personalized medicine is just figuring out, for the drugs that already exist, whether they're going to work for you or not. We can talk about that later. But then the second phase will be, you have a disease, and we're going to use in-depth information about your DNA and your lifestyle and what you eat to choose or even design a medication that is specifically focused at your particular molecules and your particular disease. This is currently the most advanced in the setting of cancer, where we can take the cancer cells out of the patient, characterize them, and say, for this particular patient, these 3 drugs look like they might work the best. And that's very exciting, and it's starting to revolutionize cancer therapy. But really, we'd like to be able to do that for all diseases eventually.
Wow, okay. Sounds exciting.
Yeah, very exciting. Keeping us busy.
And so in a lot of our audience, a large portion of our audience are students, high school students, college students. And a lot of them are interested in bioinformatics and bioengineering. And so a lot of them start learning coding, right, for computer programming in high school. And so is there a preferred language or languages that computational drug designers use?
There isn't. And I specifically try to let my students in my laboratory use whatever language they're most comfortable in, because I don't want to be dictating. However, I can say that everybody needs to know 2 languages, generally speaking. One is what I would call a prototyping language, where you can get your ideas running really fast. That would be things like Python, Perl, R, maybe MATLAB. They're not the fastest languages in the world, but they allow you to get your ideas tested quickly. Then the second language you need to know is a language that can be very fast. Because when we're talking about genomic level analyses of a genome, there's 3 billion letters in a human genome. There's a lot of measurements we can make. Once you've proven that it works with your Python script or your R script, then you go to something like Java or C or C++ to get the speed that you need, and also to run perhaps even on a cluster computer. So my laboratory, the students have laptops. They write their code on their laptop. It's just a regular laptop like everybody has. But when they get really serious, they move over to a cluster where we have like 200, 500, 1,000 nodes that can do a large-scale analysis. the key is you don't want to waste time on the cluster. So you debug on your laptop, convince yourself it's going to work, and then move to the faster machines.
And so and for our students who are listening, I mean, the proficiency with these languages, I mean, is it different than somebody who would want to write software from scratch as opposed to somebody who wants to apply functions to search for,
So in my field of bioinformatics, we are actually writing code. So there's also a lot of good work you can do in biological research where you're using codes that other people have written and made available. And that's great, and because then you can leverage all that effort. And there you're really focusing on solving the biological problem. In our lab, we're interested not only in solving the problem, but building novel methods because we know that the current codes don't work very well. So we're always on the lookout for codes that kind of work okay, but there's clearly room for improvement. That's an opportunity for us to take our knowledge of biology and our knowledge of coding, combine them, and come up with a new package that might do something better. Just like, and that really is just like all computing, right? You have your favorite texting app, and then somebody comes out with a better texting app, and if it's good enough, you'll switch. And same thing for biologists. If you give them a better piece of code, and prove that it's better, they will switch what they're using. And that just makes research go faster.
Mm-hmm. And so is there moving, the programs and softwares that you produce from the bench to, say, the clinic? Of course, yes. Most clinicians don't have computational backgrounds. So is there an emphasis in making, I guess, a user interface friendly or user friendly, form of your code or your software?
So that's a great question because almost everything is what I would call research grade. It works, it shows the idea, but you would never give it to a doctor. Doctors have like 12 minutes per patient. They're doing a lot of things. They're examining the patient, they're talking to the patient, they're filling out paperwork. If I'm going to put a new piece of software in front of them, I have to make sure that it doesn't disrupt their workflow at all, that it has a user interface that is pleasant and it doesn't make them angry, but makes them actually happy. And if you've seen some of the medical electronic computer computational tools, they are not very good. And so you're hitting on a huge, I think, opportunity for young people who learn about what we call human-computer interaction. How do people make apps that people enjoy to use? And then marry that with a deep understanding of how doctors work, because we really need user interfaces for physicians that are much better than currently existing. So that I think that's a great opportunity for young people entering the field. How do you make doing medicine with computers fun and not a pain in the neck?
So on that note, could you describe for us your academic journey beginning with high school? You're originally from New York, is that right?
I'm from New York. Yep, I went to high school on 85th Street, and it was a regular high school. I love it. I'm very loyal to it. But we learned all the basics, the fundamentals. It was not particularly oriented towards science, so we also learned things about communication skills, speaking and writing, and then of course the basic sciences: biology, chemistry, math, physics, and then of course history, English. And I just tried to pay attention, and I tried to do well. But I was in no sense totally devoted to science. I knew I was good at it, and I enjoyed it, but I also enjoyed history and English and foreign languages. But when I got to college, I went to Harvard College. I said, you know what, I definitely want to major in the science. And so I picked biochemistry and molecular biology because at the time, that major at that particular college allowed me to take the broadest amount of science. So I could get the next level of biology, chemistry, math, and computer programming, importantly. I had played around with computers in junior high and a little bit in high school. I knew that I liked them, and this is different now because I think people are learning computer science earlier, but back in ancient times when I went to school, a lot of people just got their first experience programming in college, believe it or not. So in my freshman year is when I really wrote my first serious set of programs. But I loved it. I immediately said, and people who know how to program and who love programming, they know that feeling. It's like, oh my goodness, this is really great. So I became a TA in that class to help teach it. And I said, you know what, I'd love to get involved in biological research because I might want to go to medical school. And all the pre-med advisors were saying, well, you might want to get involved with some research so you can see where does medical knowledge come from. It doesn't come from a textbook. It comes from people doing research and discovering things. So I was really lucky that I found a laboratory that needed somebody to write a program to analyze. They had images. They were taking pictures of things, and they wanted some primitive, simple analysis of those images. But it was all computational. So that got me my start. I did that, and I really enjoyed it. And then I found out that there were MD, PhD programs. So at the end of college, I was thinking of medical school, but my advisor said, well, Russ, you're kind of good at research too. Do you know that there are these programs where you can get both a medical degree and a PhD? Now, it takes 7 or 8 years, so you got to really be sure you want to do both. But I was quite sure. I was very devoted to the idea of taking care of patients as a doctor, but I also wanted to do research. So I came to Stanford University for my graduate medical school, and I did my MD for 2 years. Then I did my PhD, and then I finished medical school for another 2 years. Wound up getting both degrees, went off and I did my residency, so I'm actually a physician. I'm a general internist. But then my life has turned into mostly research with like half a day a week of being a doctor. And for me, that's the right balance. Everybody has to decide, do I want to be mostly a doctor or mostly a researcher? And I would never even dare to say what's right for any individual because you have to make that decision. So it really just started, high school, and then a little bit of focus in college on science, and then a focus on medicine and computers in graduate school, and then looking for a job. The one thing I would say is early in your life, in high school and college, you want to focus on the fundamentals, the things that will serve you forever. Basic math, basic computer science, basic biology, chemistry, and physics. You don't have to worry about the bioinformatics. You can keep that as a goal, but before you can really be meaningfully useful to those fields, you have to have the basic core knowledge. And so it's all about getting good fundamentals. I still have, in fact, on my bookshelf behind me, I have my high school calculus book, because that's when I learned math and calculus. And if I have a math question, I go to my high school calculus book. That's the one I trust.
Wow, yeah, thank you for, and I think that might be a surprise what you said to a number of our students who, of course, in their minds wanting to study, science in the future, worrying about, beyond calculus. You know what I'm saying?
Right.
Typically, the highest calculus they can take is, calculus BC, right? AP calculus BC, which is second semester calculus, right?
That's what I took. But you know what? That just comes up over and over again, and it gives me the fundamentals so that when I'm looking at a fancy new mathematical technique, I really rely on my BC calc to remember about integrals and derivatives and Taylor approximations. They come up all the time. And so same with chemistry. Chemistry in college, I have my chemistry textbook behind me on my bookshelf, and I have to remember about organic chemistry or physical chemistry. And I can't stress how much taking care of the fundamentals works, because the applications, what you're going to be using this knowledge for, changes over time, but the fundamentals don't change as quickly. So math is math, and math is not going to change in the next 50 years. Chemistry is going to change a little. Physics is going to change a little. Biology might change a little bit more. So focusing on things that won't change as much gives you something that you can kind of bank on for your whole life.
Okay, well, thank you. Well, for those of you who are joining us now, we are having an interview with Professor Russ Altman at Stanford University. His specialty is in computational drug design. So our next question is, so even successful drugs don't always work on everyone, right? How does genetic variation in people affect the process of drug design?
Well, thanks for that question, because this is my favorite topic, as you probably know. So there's a field called pharmacogenetics, pharmaco for drug, genetics for genetics. And this is the study of how your genetic background, the DNA that you got from mom and dad, and which they got from your grandparents, how does that affect your response to drugs? Most people don't realize this, but you inherit your response to drugs just like you inherit your color of your hair or how tall you are. And so we know from the last 20 or 30 years of research that there are certain, even though most humans are very similar in their DNA, there are small differences. Those small differences in DNA sequence can lead to very different responses in drugs. For example, the rate at which you absorb a drug out of your gut into your bloodstream can be very different based on the specific genes you have. Similarly, how fast your liver metabolizes it, get rid of it. Basically, when your body sees a foreign substance, it tries to get rid of it. That's a very kind of handy thing if you've eaten a poison. And in fact, your body doesn't know when you're taking a drug. It doesn't know if you're supposed to be taking that drug. It just knows that it's foreign, and so it will try to metabolize it and get rid of it. How it does that can differ across individuals, and we now have many, many cases where a drug might be great for one person, works great, no side effects, but for another person, because they have DNA differences, it might not work at all and might have a ton of side effects. That's not used in practice today, but the vision that we have is someday you'll have your genome in a database, you'll go to your doctor will say, oh, you need to be treated for this disease. Let me check the database to see which of the drugs that we have is going to work best for you. Or in the farther future, it might even be, as we were talking earlier, let me see if I can design a drug for you that'll work perfectly. Right now, we're really shooting for that first thing, which is among the drugs that we have, which one is best based on your genetic background. And so as a doctor, I'm setting up a clinic here at Stanford. In fact, we're going to kick off next month, where people will come and we will measure their DNA. We will ask them if they're having trouble with any of their drugs currently. And if they are, we'll try to figure out is it a genetic cause for that trouble. If they're not on any drugs, it will still be useful because we can tell them in the future, if you're ever on this drug, be careful because we predict you might have trouble with it. And we might even give them a little card or something so they can show that to their physician. So it's a very exciting field called pharmacogenomics. And we've been working on it for 15, 16 years, and we think it's almost ready for prime time.
Wow, that sounds very exciting. So what are some notable drugs that have been successful?
Yeah, so let's see. There's a bunch. Let me try to pick one. Of course, hopefully most young people are healthy and not on medication. Some of the people listening might not be, but they probably have parents or grandparents who are on drugs. One would be a drug called warfarin, which is a blood thinner. And you take that drug if you've had a clot, either a clot in your leg or in your lung or in your heart. And it turns out that warfarin is incredibly difficult to get the right dose because it's not the case that big football players get a big dose and little tiny people get small doses. It's totally unpredictable from your size. It has more to do with your genetics. So we've written some papers showing that with genetic information, we can get a precise dose of that drug on the first try, as opposed to having the patient come every week and adjusting their medication to try to get it right. So warfarin is one example. Another one would be antidepressants. Depression is a huge problem in our society, and we don't treat it very well. But there are some antidepressants that are metabolized differently based on your genetics. And so there is now a lot of knowledge about how to choose antidepressants based on your genetics to try to maximize the chance that it'll be effective. And then, of course, for somebody who's really depressed, there's a sense of urgency to get them feeling better as soon as possible. And so any time you can save at getting the dose right or even picking the right drug would be incredibly valuable. And we have a list, I think, of about 30 or 50 drugs now where there's really good evidence for the genetic influence. And it's not that there's just 30 or 50 total, it's just that's the one where we have enough data to really act on it.
Okay, wow, that's exciting. And so
really, that's what I'm hoping to focus on in my clinic. Those 30 to 50 drugs are ones where there's a lot of good evidence, and so I can really make recommendations to the patients and to their primary care physicians. I really think you shouldn't be on this unless it's working great for you and you're not having side effects. It just looks to me like it might cause trouble.
And so for people who are listening and who might be interested in being part of that trial or study, can they just walk in? I mean, do they sign, what about, I guess, personalized information? Things like that?
Yes. So there are a lot of research studies looking at pharmacogenomics. There's some in the Nashville area at Vanderbilt. There's some at Chicago, at the University of Chicago. The clinic that I'm describing is actually it's not a research, it's going to be an actual clinic where it's going to be part of their care. And we're going to have initially a very small, I want to start small, so it's going to be by referral only from my colleagues at Stanford Hospital. That's not because we want to close it out to people. It's because I want to make sure we're doing a good job before we open it up to a larger group. So it's going to be a couple of patients a week, make sure we take good care of them, make sure there's nothing bad happens. And then if that's successful, which I'm hoping it will be, then we'll open it up and make it available more generally. And we will publish our experience, even though it's not a research, we can still write about it. And that might make other doctors feel emboldened to start opening up their own clinics in this area.
Wow, okay.
But if your listeners are in high school or college, they should be able to get pharmacogenomics in their lifetime for sure.
Wow. Okay, that's exciting. Wow, I was going to ask you how far off do you think it's going to be in terms of getting to the clinic as a something that Kaiser would cover, so to speak, right?
Right. So one of the main issues is getting insurance companies to pay for it. Right now, they're a little bit waiting to see if it's a good thing, if it helps patients. So right now, in my clinic, for example, people are going to have to pay cash for the genetic test. It might be a couple $100. For some people, that's not a problem, but for other people, that's a lot of money. I totally understand. And so if we can show the insurance companies that this is really helping these patients, then maybe they'll pay for it for all of their covered customers, and that'll make it more generally available.
And do you ever have, the concern of, the movie Gattaca, where everyone is genome sequenced, and insurance companies, of course, they would be interested in that, knowing risk. They want to calculate risk, right? And so how does a clinician or a clinic, right, that actually, gets people's DNA, right, and stores it, and communicates with insurance companies, how do you balance that?
It's a great question. We have to really make sure that people's genetics are not used to hold things against them, like to deny them employment because they might have a higher risk for some disease. The key thing so Gattaca is a great movie, but it suffers from a major flaw, which is it acts as if everything is determined by your DNA. And in reality, DNA is just adjusting probabilities, but it's not determining things. So it might be that the average person has a 1% or a 5% chance of getting colon cancer, but because of their genetics, it goes up to 10% or 15%. It still doesn't mean you're gonna get cancer. And what are the other influences? It's how you live your life. Do you exercise? Do you eat right? Do you go to the doctor and have preventive medical care, like all the screens that they do? So the mistake that Gattaca makes is it overemphasizes genetics as determining everything. And genetics determines some, but it's more like changing the probabilities. So that's the first thing I wanted to say, is that people shouldn't think that genetics is just giving them a readout for the future. It's just adjusting probabilities. Now, in terms of the data being used in bad ways, we definitely are focusing on how to create secure databases where only credentialed people can get to them and use it on behalf of the patient. My belief is that the patient should decide who gets to see their data. We should build systems where if a doctor needs to see, on your smartphone, maybe you type in a passcode that says, this doctor is cool and this doctor can see my data. Maybe in the emergency room, you loosen that up because maybe you're not able to communicate. But we can deal with that. We can figure out systems. So I'm a big believer that patients should control who sees their data. I think that'll be a great day when that's the case. It's not there today at the hospitals and the clinics. I really would like to see it on the cloud under the control of the patient. So that's another opportunity for young people to think about how do we build that world so that we have these things available to all the doctors who need to see it, but under the control of the patient. There is a law, I have to mention, called GINA, G-I-N-A, the Genetic Information Non-Discrimination Act. And that is a nice law that was put into effect, 5 or 10 years ago, that says you can't use people's genetic information against them. Now, there's still some loopholes. It's not a perfect law, but it is a good start to try to protect people from really having unfair things done to them just because their probabilities are slightly different than others.
Wow. Okay. And so our next question is how do, switching gears back to more molecular topics, how much do changes in protein conformation affect drug design?
Wow, that's a very technical, that's somebody who really knows something, obviously. You obviously have scientific training. So just to remind everybody, proteins are the molecules that are encoded for by the genome. So the genome has genes, and they get transcribed and then translated into these beautiful three-dimensional molecules that you may have seen pictures of. And those are often the molecules that our drugs bind to and modify their function. So your question is really, as these proteins are not frozen into a single structure, they actually are kind of living things almost that move around and can change their shape. And that's a huge issue. It's a huge issue for our research. And one of the areas that has led to is the research area called molecular dynamics, where we use computers to simulate the motions of the proteins so we can understand how much they're going to be moving and how that might affect or change how we want to design a drug. And I'm very proud that some of that work was done by my colleague here at Stanford, Michael Levitt. He was one of the founders of this field of molecular dynamics, and he won a Nobel Prize for it 2 years ago. And of course, that was very thrilling for everybody who knows him. He is a great guy and very deserving of this award. And his contribution was figuring out how to use physics and computers to understand how proteins move and how their shapes change. So we're pretty good at that, and therefore we can use that information when we're designing drugs.
Wow, yeah, okay, maybe we'll try to get him on the show too. So, so at this point, we're gonna have a fun segment in which we invite you to, if you want, do any fun, humorous trash talking to any competitors or colleagues in the field. We'd be happy to invite them on the show to respond to you. So, feel free to, if something comes up, just stop me and, we'll give you your spiel, okay? All right, so.
Yeah, go for it. What should I do?
Well, if you want, if nothing comes to mind, then it's okay. If something
there's no trash talk. Let me just say that people in science tend to be a pretty good community. It is very competitive, but we're all kind of focused, especially in biology and medicine, on human health. We do get very competitive to be the first to show something or the first to do something. But no, of course, I'm a big fan of Stanford, and I love the scientists here. You may have heard in the news that we're part of this very exciting initiative that Priscilla Chan and Mark Zuckerberg announced last week. It's a big effort to try to, believe it or not, cure, prevent, or manage disease in the next century, by the end of this century. And they've put up big-time resources, and they're getting us to work at Stanford with our colleagues at University of California at San Francisco and Berkeley. And so that's really fun because we have 3 times the number of colleagues involved. And I think that's a great example of how people from industry who've done very well can kind of give back to their community by creating these big, what I would call like a moonshot project that galvanizes all the scientists and gets us excited about trying to really cure all disease, which is sounds like a ridiculous goal, but if you listen to Zuckerberg and Chan talk about it, they really did their homework and they really believe this is possible. So it's incredibly inspiring, and I'm happy to be associated with it.
Wow, yeah, that's very exciting.
That's as close to trash talk as I can get today. Sorry, that doesn't sound very much like trash talk, does it?
Well, it's positive trash talk.
Yeah, but it's great that a guy who built Facebook is turning around now and saying, let's cure disease. That's really awesome, and he deserves a ton of credit.
Definitely, yeah. So, there's a joke in the sciences that, theoretical physicists feel that they're at the top of the totem pole until they catch a cold. Then they go to their...
Exactly. I do all this research, but if you ask my mother what I do, she'll tell you he's a doctor.
All right, doctor. Well, the next molecular technical question is, are there certain categories of proteins that are harder to create drugs for than others, and why?
Yes. In fact, this is a big challenge. So it's easier to talk about the ones that are easy because there's 4 types of proteins that we're really good at making drugs for. One is called G-coupled proteins, which are receptors on the surface of cells, and we have a lot of drugs against those. In fact, 30 or 40% of all drugs work against this family. Secondly, we have something called nuclear receptors, like the way you process androgen, testosterone, estrogen. Those are very, what we call druggable. Ion channels. These are also cell surface molecules that allow ions to go in and out of the cell, and we're pretty good at modulating those with drugs. And then something called kinases, which are these proteins that add and subtract phosphate groups to molecules, and they're very important for cellular signaling, telling the cell, we just sensed some problem and we need to change what the cell is doing. So those 4 are very easy to get drugs to, those 4 types of proteins. But there are many, many others that we would love to have drugs to that we just haven't been successful. And that's where computational techniques can be useful. And in fact, we published a paper, I think, a year ago showing a new way to evaluate whether a drug might be possible for a difficult to drug protein. So it's a great question and a big area for future research.
All right. So let's switch gears a bit now and go asking our questions on a more, organismal level as opposed to molecular level.
Yeah.
And so drugs are foreign chemicals, right, that can be toxic to normal cells too, not just disease cells. So how does a drug designer balance the ease with which a drug enters the cell and the ease with which the cell can remove that drug?
That is a great question. What we often see, and just to support your question, is we often see a first-generation drug, like the first of its kind, usually has terrible dosing. Like you have to take it 4 times a day. Anybody who's ever tried to take a medication 4 times a day, it's incredibly difficult. We're all living our lives, and we're only awake for whatever, 18 hours, 15 hours, however much sleep, and like 4 times a day is very difficult. That's because sometimes, in order to get to work the best, you have to get in and get out very quickly. But patients hate that. So very often, the second and third generation of these drugs will be once a day, or at least twice a day, and then eventually you'd love to get to once a day. And that all has to do with the transporter molecules on the surface of cells that actually grab the drug and pump it across the membrane into the body. And the only way to manipulate that, really, is to have a chemist think about how those transporter molecules work, and think, how can I modify this medication so that it still does what it's supposed to, but it goes across the membrane a little bit more slowly, or sometimes a little bit more quickly. And a lot of it is trial and error, but the drug companies are very good at doing that because they know that what people really want is a once-a-day drug. And so you have to make those chemical modifications and try them out until you hit the sweet spot, so that the drug is essentially coming in and getting eliminated within 24 hours, so you can wake up the next morning and take your next dose and everything's going to work fine. So it's really about chemical manipulation.
Okay. The next question is, how then does a drug designer send a drug only to an organ or certain cell types in the body?
So I must say that in general, we don't do that. So when you take a medication, for the most part, it goes everywhere in your body. That often explains a lot of the side effects. So, you mentioned before nausea or vomiting or a headache. So I'm giving you this drug maybe because of what it's going to do to your liver, but some of it gets to your brain and causes a headache. So there are people working very hard, and you might have heard, and you might have even interviewed people in the field of nanotechnology. But there's now an idea that maybe we can add molecules to the drug that will carry the drug preferentially to its target organ and then release it. So that's super cool and new wave, because now I don't have to worry about it achieving very high concentrations in the brain and causing a headache, or achieving high concentrations in the gut and causing vomiting, because I can put it as like payload on a targeted molecule that either perhaps using antibodies or using other technologies will just go to the organ of interest. So that in general is not happening, but we're starting to see it in cancer, where what they're doing is they're saying, let's figure out how to deliver cancer drugs only to the cancer cells and not to the rest of the body. So I would count that as a big area for research, and one that we're going to see more and more of, because it's a great way to handle these side effects.
Okay, and so, on that point, so kidney damage seems to be a common problem with many drugs. So why is that, and why hasn't that been solved?
Yeah, so that's a great question. So there's 2 things about the kidney that you have to know to understand this. The first is the kidney sees way more blood flow than it needs to live, because it's a filter. And so an amazing fraction of your total blood flow, like per heartbeat, is going through the kidneys. So first of all, the kidneys are seeing a lot of blood. Second of all, and therefore if there's toxins in the blood, the kidneys are seeing more of it than other organs, because they're seeing more blood per minute or per second, or however you want to compute it. The second thing about the kidney is those cells are incredibly sensitive to toxins. They die easily. And of course, I didn't design the kidney, so I'm not responsible, but it's a fact. And so the combination of seeing a lot of blood, and therefore a lot of foreign chemicals and maybe even toxins, and the fact that they're very sensitive, means that if you're going to have a problem with a drug, the kidney is the first place you might see that. Not for all drugs, but for many drugs. And so we were talking about animal testing earlier in our conversation. One of the things we look at very carefully is how are the kidneys of the mice and the rats doing when they see these drugs. Because if we see signs that the kidney cells are dying in response to the drug, that means that drug is in trouble, and it's probably not going to make it to the next phase of testing.
Okay, as we come to the end of like Q&A segment between me and you, can we end, just for our audience, high school students, it's easy for them to be inspired by someone like you. But it's also easy for them to be intimidated, right? And scared away for somebody who knows genetics, who knows biology, who knows biochemistry and computers. And so, were you always that way? How long did it take you to become comfortable, right, in these areas?
It's really a lifetime. This is why I made it so. I still don't feel like I know all the things that I could know about any of those fields. So what do I do? But I don't want to go crazy, and I don't want to feel like I'm terrible all the time. So that's why I made that comment earlier about focusing on fundamentals earlier in your career. Make sure you learn the math, learn the chemistry, learn the physics, learn the biology. Don't worry about the applications. Second of all, we all don't know exactly what our pathway is going to be, and there's many, many pathways to being an excellent contributor to society. So given all the choices, I really encourage young people to follow whatever their passion is. And I'm sure they've heard this before, but it's really true. You're not going to be good unless you love what you're doing. So never do something because somebody else told you to do it, or somebody else said that this is the way to go. You have to go with your instinct about, A, what am I good at? And B, what do I enjoy? And if you're really lucky, those two things will be the same. I'm good at stuff that I enjoy. That's typically the case because, of course, we enjoy it more. So you have to be aware of what you're good at, and you have to be aware of what you really enjoy, and you have to pursue your passion. Don't think that anything is out of bounds because there's probably a way to get to where you want to go, and you don't have to have the full plan. You just have to have the next step, and that makes it way less scary. It's scary to say, how do I become a professor at Stanford starting out as a sophomore in high school? I have no idea what to tell somebody. But as a sophomore in high school, you want to do well as a sophomore and make sure you get to junior year. That's doable, okay? So if you take one step at a time and you try to be excellent at each step, then good things will happen. So I try to take pressure off young people by saying you don't need a master plan, you just need your next step. And the next step is a combination of hard work, loving what you do, and being passionate about it. That's the best kind of advice that I could give, I think. Hopefully that is accessible and makes sense to people.
Yeah, definitely. I think that's great advice. In the future, we plan to have a roundtable discussion with professionals about the question, should a student pursue passion or practicality? Maybe you'd be interested in joining that discussion.
Yeah, that's an easy one for me. Forget practicality. Because if you hate it, you're going to suck at it, and then you're going to be doing something else. I'm done. That's my whole speech for the panel, so you don't have to invite me.
Okay. All right, Alistair.
Okay, we do have a couple of questions. I'm going to read them off to you, Dr. Altman. The first one is from Sarah Yang. She asked, what's the workload like when you're in the PhD and MD program?
Yes, the workload is very high. That goes back to what I just said. It would have been terrible if I didn't love it, but I loved it. So during graduate school, I was able to get married. I was able to have one child. I went on to have 3, but I had one when I was still in my PhD. It made my PhD go a little bit longer because I remember writing my PhD, the actual document, with the baby asleep. And then when he woke up, I had to go outside the building so I wouldn't bother everybody and play with him. And if he fell asleep again, I came back in and started working on my thesis again. So it is very hard work. But I mentioned my marriage and my kids to show you that it doesn't mean that life has to stop. It just means that you have to make choices about how hard you're going to work. And sometimes I had to say no. Sometimes I had to say, I know I could work harder, but I'm not willing to work any harder than this because it would start to impact my happiness with my family or with my friends. And everybody has to make that decision because you can always work harder, but at some point you say, but I'm only willing to do this much because after that, it's not fun.
Can I just interject a question? So, and again, for our student audience, Dr. Altman, were there times when in your training throughout when you felt inadequate or wanting to give up and, you just overwhelmed?
Yes, let me tell you that story. It's very important. So I joined a lab that was not really a computational lab. I thought initially when I was a first-year graduate student, I was going to be in this project that was very exciting, I thought, but it required test tubes and not really a lot of computation. And I was terrible. I didn't mix my buffers right. The pH was supposed to be 7.4, and I could only get it to be 7.3, and that's a huge difference. You'll learn in chemistry. And I did this for about 3 months, and I said, you know what? This is not working, and I'm afraid that I might be a failure. And in fact, I'm afraid that maybe Stanford made a mistake, and they shouldn't have accepted me because I'm not really, I looked good on paper, but I'm not really that very good graduate student. And so I went to my advisor at the time, and I said, you know, I must say, I'm thinking that this is not right for me. And he said, you know what? I think you might be right because this is not your sweet spot. And when I read your application, they were all talking about how great you were with computers, and they said you were okay in the lab, but you're not even really okay. You're worse than okay. So that was a very hard time. And what I did is I switched labs. So I lost those 3 months. Those 3 or 4 months were gone. But it wasn't wasted because I learned something about myself. And it's where I get this advice about do what you're good at, because I was not good at that. And it would have been a disaster if I continued. And fortunately, a combination of myself, my friends, and this guy helped me realize that and get back on a track. I joined a new lab. It was computational. It was what I was good at. Within 2 weeks, I had made a contribution. It was a tiny contribution, but it was so important for my confidence that I was able to do one little thing that the new lab found useful, and then I was off to the races. So yes, you have to persevere. And these negative things will happen, and you have to take a deep breath, learn from them, and adjust. Thank you, Sarah and Alistair, for those questions.
We got one more question. I know you have to run soon. This last question is from Justine. She asks, do you think pharmacogenomics will be brought to the masses soon? And also, is this major even offered in colleges at the moment?
So let me answer the second question first. Pharmacogenetics is an advanced topic that you would need genetics and pharmacology. So it wouldn't probably be appropriate for an undergraduate major. What you would probably do is chemistry or maybe genetics and biology. In fact, even genetics is sometimes not even a major, so it might be biology. So that's what I meant by fundamentals. You have to walk before you run. So if you're interested in pharmacogenetics, figure out if you want to do chemistry and druggy stuff first and then learn the genetics later, or do the genetics and the biology first and then you'll get to pharmacogenomics and genetics later in your career. I think it would be jumping the gun to be a major in something like that before you get the fundamentals. So that's the second question. First question, well, I wouldn't call it the masses because in my clinic, we're going to start seeing 2 or 3 patients a week. So that's not mass. But if we're successful, we're going to try to train other doctors to use this technology. And so I'm hoping that in something like 5 years, and I would be disappointed if it wasn't true in 10 years, any physician or patient who wants to use genetics in prescribing drugs would be able to, and it would be somewhat routine. So give us 5 or 10 years, and you can hold me to that and send me an email in 10 years if you're still unable to get pharmacogenomics from your doctor.